started. First, welcome everyone to another installment of our Diatom Web Academy. It's brought to you by the Society for Freshwater Science Taxonomic Certification Committee and Diatoms of North America. And we've been meeting every couple weeks for a couple years now, bringing us together as a community, trying to find ways that we can continue to connect even in times that Zoom meetings have become a little too normal for all of us at that. The webinars are, of course, always recorded. You can find the information at diatoms.org/news, and also the links to the recordings. Most of the recordings are posted within 3 days to a week after the previous one. I have just a couple of announcements to make. First, we're going to have another Web Academy in 2 weeks by Dr. Joe Mohan, who's just finished up his doctorate at the University of Maine. He's going to be speaking on diatoms from the third dimension, integrating 3D computer modeling into diatom research. That'll be at the same time, again, 2 weeks from now on March 15th. I also want to provide a little information, talk to one of our colleagues, Bob Pillsbury, who's at the University of Wisconsin, Oshkosh, and he has a 2-year fully funded master's project working on Didymosphaenia in Lake Superior and connecting waterways. I'm going to put his email and his address in the chat. That's a 2-year Master of Science program with fully funded at University of Wisconsin in Oshkosh, working with Bob Pillsbury. Please reach out to Bob if you know of anyone who is looking for a master's degree, and I believe it would be starting as soon as this fall. All right, without further ado, let me introduce today's speaker. Matt Ashworth is joining us. He's from the Department of Molecular Bioscience at University of Texas in Austin. However, he is not joining us from Texas. He's actually in London visiting the British Museum right now. And so he's joining us from London. He's going to be speaking on Biddulphia or Odontella and giving us a guide to the morphology of these benthic marine mediophyte diatoms. Matt, you go ahead and pull up your presentation in full screen and we'll get started here. Thank you everyone for joining us today.
Okay. Thank you, Mark, for the introduction, and thank you all for showing up, listen to me ramble about the Biddulphioid diatoms. So this is a group of diatoms that I've been working with for a while now. They take good pictures. They are not particularly well studied, but hopefully by the end of this talk, you'll have an appreciation of their diversity as well. So I've already spoken too much. Let's move on. All right. So what do I mean by the mediophyte diatoms? Well, the mediophyceae, dang it. The mediophyceae are this fairly poorly described group of, well, really it's a series of groups of radially symmetrical diatoms with distinctive apices that grade into the bilaterally symmetrical diatoms. So when I say distinctive apices, I mean we're going to be seeing a lot of corners here, right? So a lot of boxes, like with this Lithodesmoides, Trigonium, the setae of Higgocerus, these long linking spines of Hemiolus, and then however you classify something like Isthmia minima. If you've taken the diatom course at Iowa Lakeside Labs, you've seen at least a few of these. Things like Higgocerus and Ursulenia, and then Pleurocyra and some of the thalassiosauroids, so Stephanodiscus, Cyclotella, Cyclostephanos, those sort of round, radially symmetrical planktonic diatoms. Now, of course, that just is a drop in the bucket when it comes to mediophycean diversity. A lot of this diversity is in marine waters. And it's not just taxonomically diverse, but it's pretty morphologically diverse. So, again, a lot of corners, bipolar diatoms like these Biddulphia, the Somatocyreales, the Eudotogramales, some tripolar diatoms like this other Biddulphia, or a Triceratium, I'm sorry, a Pseudictiota, or even some long and bilaterally symmetrical diatoms like Artisonia, like some of the Artisonias. So there's a lot of diversity in here, morphologically and genetically. So I mentioned there are a couple of types of these distinctive apices. I'm primarily going to be talking about the Biddulphioids, which have sort of distinct elevated apices. Now, there are a couple of mediophyte groups that have similar structures, like the hemioloids, things like this Carodolina or Hemiolus, also have these very long apical elevations. But the difference between these hemiaelian elevations and those of the Biddulphioids and the Eupodiscoids has to do with the form of attachment, right? So with the hemiaeles, we have these physical siliceous attachments, these actual linking attachments on the frustule, joining the cells within a colony. Whereas in the Biddulphiales and the Eupodiscales, attachment to the substrate and attachment to the colony is typically done through secreted polysaccharides, this EPS connection, which will be secreted from elevated pore fields. So the Biddulphiales, we've got attachment through these secretions through pore fields. Hemiaeles, physical attachments through siliceous structures on the valve. So we're going to be talking about Biddulphioids here today. So even within the Biddulphioids, we have a lot of variation, both in the morphology, the gross morphology, the outline of the valve, ranging from circular to bipolar, tripolar, and beyond. But we also have quite a bit of variety in the finer valve morphology, both in the structure of the apex, the valve apex, and the construction and ultrastructure of the valve itself. So historically, a lot of the classification of these taxa has been done based on the gross valve outline and on the apical morphology. But that still left genera with a lot of morphology within a genus, a lot of morphological variation within a genus. So, come 1971, Ross and Sims decided to have a look at the Biddulphioids with the scanning electron microscope and really tear into and describe the fine structure of not just these elevated apical pore fields, but also of the rest of the valves themselves. And in terms of the apical pore fields, they described a couple of different types. The two main ones we're going to discuss here are the ocellus and the pseudocellus. So the ocellus, this is an elevated plate of silica, normally surrounded by a thickened structureless rim, pierced by these closely packed holes. So a very definite, distinct difference in the size and density of the pores on this ocellus as opposed to the rest of the valve. As for the pseudocellus, we still have an elevation, we still have a change in the size and density of the pores, but there's no strong demarcation. There's no rim surrounding it. Now, in addition to the apical pore fields, they also looked at the valve itself and the cross-sectional ultrastructure of the valve. So some of these Biddulphioids had porous valves, where the pour through the valve. It's more or less the same diameter all the way through with an external pore occlusion. But they also found a number of loculet valves. Now, in a loculet valve, we have this two-layered valve where there's, excuse me, I'm already not used to talking this long. There's actually a change in diameter in the pore size as you go through the valve. So there's an actual chamber or locule within the valve itself between the external and internal openings. And in a loculate valve, the external opening will be occluded by some form of cribra or other type of vellum or pore occlusion. Now, they also described a third type of valve cross-section. So there's also chambering in pseudoloculate valves, but with these valves, there's a basal porous layer that has the chambers actually external to the valve surface, and then these foramina open to the exterior of the valve. So no occlusions on the exterior side, and this basal porous layer on the barrier between the external and internal valve. This is something that we don't just see in the Biddulphioid diatoms, Stephanopixis. This is another diatom where you see this sort of valve cross section. All right, so we've got those basic categories. There's a couple more features that I'm going to discuss in relation to these genera today. So these are all things that you're probably familiar with if you've looked at diatoms at all, but just for a quick refresher. So there's a lot of variation with the Rhemoportula across these genera, both in where they're located on the valve, in addition to both their external and internal morphologies. The annulus, while most annuli in the radially symmetrical diatoms are circular or ring-like, in the Biddulphioids, we also see these, and in the mediophytes in general, we also see these elongate annuli, elongate or lanceolate in the case of this Higgocerus. And then costa, we'll be talking about costa a lot, these secondary siliceous thickenings, either on the exterior of the valve or the interior. So at the very least, with those basic characters of the valve cross section and the apical pore field morphology, Ross and Sims grouped the genera into four categories. So porous valves and pseudocellus. So this would be Biddulphia, loculate valves and a pseudocellus, Trigonium, porous valves with an ocellus. So these would be Odontella, Amphitetris, and Amphipentus. And then loculate valves with an ocellus, like Higgocerus and Keratolus. Now, back in 2013, we looked at a molecular phylogeny of some of these Biddulphioid taxa to see where the molecular phylogeny corresponded to some, if it corresponded at all to some of these morphological categories from Ross and Sims. And what we found is while pseudocellate taxa tended to pop up all over the mediophytes, when it comes to an ocellus-bearing taxa, ocellate taxa were monophyletic. This was a single group of ocellate taxa. This included both the porous and the loculate valves, and the porous and loculate diatoms or loculate taxa were not necessarily monophyletic with respect to each other, but the ocellus-sparing taxa, at the very least, were a monophyletic group. All right, so looking at some of these other taxa, when it comes to the pseudocellate taxa, probably one of the most well-known genera is Biddulphia. Biddulphia comes in a couple of broad morphological categories. This first one we're going to talk about here is associated with the genera type, So Biddulphia biddulphiana. This has internal costae, as you can see here. It can be a either elongate or circular annulus, but the remoportula, if they're present, are associated with the edge of that annulus. Now, the remoportula can be a little tricky in this group because there can be tubes, there can be external tubes without remoportula. Mary Ann Tiffany, back in 2015, wrote a paper that described some of these occluded tubes on the surface of Biddulphia, but some of the tubes lead to remoportula and some of them don't. Now, the mantle of this taxon is also deflected. It has this flange on the mantle. And then the valvicopula extends up and over this flange. And in the case of Biddulphia biddulphiana, there is even a dorsal extension on the valvicopula that comes up and envelops the costa. So, good way to identify the morphotype, even from the and the species, even from the valvicopula. Now, the Tridens morphotype. You see these in flora also identified as tuomii, Biddulphia tuomii. Navarro in the 90s synonymized Biddulphia tridens and Biddulphia tuomii. So I will just be referring to it as Tridens here. Now, rather than costa, the sectioning of the valve in this group are sulci. They're actually folds in the valve surface itself. So rather than a secondary growth of silica, it's the valve surface itself that goes deep down into to the mantle and creates these costa-like sectioning in the valve itself. Now, associated with these sulci, we also have an interruption in the mantle flange. So interrupted mantle flange, Biddulphia tridens, complete mantle flange, Biddulphia biddulphiana. Now, the third morphotype for Biddulphia is some of the triangular morphs. So Biddulphia alternans, Biddulphia sculpta, Biddulphia seychellensis. Alternans and sculpta are two of the most common forms you see in North America. Pretty easy to distinguish from each other in the presence of costae in Biddulphia alternans. And they're pretty different from the other Biddulphia in a couple of other features as well. Rather than being associated with the annulus, the labiate processes are subcentral to marginal on these taxa. And the internal morphology of these remoportula are very different as well. They're more stalked than you see in the Tridens and Biddulphiana forms. They're also pseudoseptate, so there are pseudosepta at the apices, and instead of the mantle being flange on the mantle, the mantle is straight and undeflected. Hydroceramides and Taxonomy, these should be familiar to those of you looking at freshwater assemblages. These are two genera which are ultrastructurally very, very similar to each other. They both have internal and external costae. They are both pseudoseptate. They both have this very distinctive undulate slit on the labiate process. And the only way they really differ is based on basically the number of apices and the location of this labiate process. Now, there is one Terpsinoe that I do want to bring up because it's a little different, and that's this Terpsinoe americana, which you might be familiar with. Now, Terpsinoe americana is as different, it's actually more different from any other Terpsinoe species as Terpsinoe is to Hydrocera. There's no undulate labiate process. There is no labiate process at all. And really, I would argue it's not even pseudocellate. If you look at the apices here, the density of the pores increases as it goes to the apex, but the size doesn't. They're closer together, but the size, you don't see that gradient in size like you see in other pseudocellate taxa. Now this also has a different pore type from Hydrocera and Terpsinoe. In particular, it has these conically occluded pores. So some of these spines you see on the valve surface are actually complete pore occlusions, conical pore occlusions. All right, Trigonium. So this one is fairly straightforward. This is a loculate pseudocellate taxa, very clear pseudocelli. You can definitely see this gradient in size and density from the apex to the rest of the valve. If you look at the loculi, you notice the external covering. All the pores are restricted to the edges of the locule there. Labiate processes are central, typically around the ring. And we also see pseudosepti at the apices here. Terpsinoe arcticum is probably one of the more common forms you might see on the northwestern and northeastern coasts of the U.S. It's a triangular and quadrangular form. All right, so Odontella. This is a genus that's had a lot of species move in and out of it over the years. In 2018, Sims et al. had a good look at the genera type, Odontella Agardh, and published a sensu stricto definition of Odontella, which included a central labiate processes, 2 types of areli. So you have these sort of cribrate forms and these papillose forms, flange on the mantle edge, and then some of the species in this group do actually have external costae, things like Odontella rostrata. Other porous genera, Amphitetris, Amphipentus. The name might suggest a very obvious way to differentiate these taxa, but you don't always see 4 apices in Amphitetris. You don't always see 5 apices in Amphipentus. Similar in a lot of ways. They're both flanged. They both have 2 types of areolar coverings. But they differ in the external costae that you'll see in Amphipentus species that you don't see in any of the Amphitetris. Now, Sims et al. Also pulled a couple more species out of Odontella. One of this was Hobaniella. So you might have recognized this as Odontella longicurus, now Hobaniella longicurus. And while this shares a lot of characters with some of the other odontelloids, we've got the 2 areolar types, although the papillose areli are very, very small and reduced. But both the external labiate process tubes and the ocellus-bearing elevations, very, very thin, very, very long in relation to the central elevation here. And we do have a flange here, although it can be reduced from some of the other Odontella species. Pleurocyra. So here's another taxa that you might be familiar with from the Iowa Lakeside Labs course and freshwater work. So this is loculate. Sort of tough to tell from the interior and exterior, but if you look at a cross section, there are locules in there. The labiate processes, if they have external tubes at all, they're very, very short. They're central to subcentral. Very slight flange to the mantle. And you might be familiar with a couple of different morphotypes. If you've just looked at Pleurocyra in the freshwater, you'll see these flat valves, these very, very flat valves on the cells. And this is the Lavis morphotype. But if you work in marine waters or estuarine deltas, you might also see this polymorpha morphotype, with a much more undulate valve face and elevated ocelli. Now, this has kind of a checkered taxonomic history because polymorpha and lavis forms have been found in the same taxon, in the same samples. People have been reticent to identify polymorpha as anything other than a form. But actually, there's been some really neat work coming out of Japan recently. Shiho Kamakura of Fukui Prefectural University presented at the International Diatom meeting and the Molecular Life of Diatoms meetings last summer on Pleurocyra lavis and the effects of salinity. So she found that if you take a culture and expose it to a gradient of salinity, it will change from the lavis form in fresh water to the polymorpha form as you increase salinity. So really elegant series of experiments, and I expect we'll see that in press soon. All right, Higgocerus. So Higgocerus is another loculate Odontella. So this is where we also start to see some of these elongate annuli, kind of like Trigonium. Rather, you'll see that the pores on the outer covering are restricted just to the edge, sort of outlining the locule. Valves are typically covered with these little branched spines. They do have a flange. The labiate processes, the remoportula, are typically marginal. They're by the ocellate processes, or they can even be on the mantle. Keratolis, whoops, another loculate diatom here. So Keratolis, from a gross morphology standpoint, is pretty easy to tell apart from Higgocerus because of the contortion to the valve. But if you look closely, you can also see a elongate annulus here. And if you look closely at the pores, you can see that the locules, rather than pores being just restricted to the edge of the locule on the occlusion, the pores cover the occlusion entirely. And then we also see a new structure here, rather than just a flange on the mantle. We see this secondary growth on the flange, which can actually grow together and enclose to create a tube, a hollow tube that goes all the way around the mantle of the diatom. What Sims et al. Termed a Scyphio marginalis. All right, another loculate genus, if you're familiar at all with some of the marine plankton. So this is Triaris. So Triaris, we don't get that contortion that we see in Keratolis, but we do still see the same sort of areolar occlusion with the porous plate completely covering each one of the locules. The external tubes of the labiate processes are very tall on the external tube, and they're often associated with the valve margin and this distinctive sawtooth or serrated ridge that follows along the margin of the valve. Some of the more common species on the U.S. coast vary primarily in the relationship between the labiate and remoportula, between the remoportula and the ocellus. So Triaris mobilensis, where they tend to be a little more distant. Regia, we start to see a much more close relationship between the two. And then Triaris sinensis, where the ocellus and the labiate process actually share an apical elevation, which you can also see in the live cell here. Okay, so if you've been paying attention, you probably have noticed that I have not mentioned Triceratium, nor have I mentioned the pseudo-loculate valve cross section. So yeah, Triceratium is another genus that's had a lot of taxa added and subtracted over the years. But Ross and Sims restricted Triceratium to these pseudoloculate, the pseudoloculate valve construction, where you've got the basal porous layer, and then these external locules on top of that. So Triceratium has marginal remoportulum, and like Keratalis, it also has a Scyphio marginalis, as you can see in this incomplete valve here, this Triceratium robertsianum. Now, this is distinguished from some of the other species within Triceratium by the valve cross-section. So in 2018, Sims et al. Noted that some of these Triceratium species did not have true pseudolocks. Rather than a basal porous layer underneath the external locules, they just had pores. That is an irregular assortment of pores with pore occlusions. In fact, two types of pores again. So they described this group as Pseudictiota. And this also has a flange on the mantle, as opposed to a Scyphio marginalis. And some of the common taxa that you see in the US would be Pseudictiota dubia with this tripartite pattern, or Pseudictiota bicorn with a quadripartite pattern. Either way, in both Pseudictiota bicorn and dubium, you'll see that the labiate processes and the ocelli are on sequential elevations. Another false pseudoloculate taxon, series of taxa that Sims et al. 2018 described was this Ralphsiella. Some of these species, within those false pseudolocks, there's only a single pore of one of two morphologies. Again, not the basal porous layer of the true pseudoloculate morphology. So this includes things like what was formerly Keratalis smithii, this circular taxon here. You have a look at the ultrastructure, obviously very, very different from Keratalis sensu stricto that we illustrated earlier. So smithii, and then Ralphsiella minima, which you'll see on the US coast on occasion. Now, minima, if you look at it in the light microscope, you might mistake it for Higgocerus because of all the little spines covering the valve. But if you remember Higgocerus, where, I mean, obviously it's loculate, but that's not always obvious on the light microscope. But Higgocerus had a elongate annulus, and Ralphsiella will have a circular annulus. So that's one way to tell them apart in the light microscope. All right, some of the other circular forms, things like Eupodiscus. So Eupodiscus is loculate, and it has pores all the way across the areolar covering, like Keratalis and Triaries. Eupodiscus does have a Scyphio marginalis, which you can sort of see at the edge here with the incomplete silicification. Then there's Pseudoliscus, which is also loculate, but like Higgocerus and Trigonium, the pores on the external occlusion are just restricted to the outside, the edge of the locule. And again, this can be distinguished from Higgocerus in the ring-like annulus as opposed to the elongate annulus. Eliscus, if you see Eliscus, you know Eliscus. It has these very distinctive external costae and sort of external arrangement of pores. It does have a Scyphio marginalis. I'm actually not 100% sure of the cross-sectional morphology. I've never seen a broken valve. But Mastodiscus, which phonetically somewhat similar. This one is definitely porous. None of those external costae, but there is sort of a raised ring structure around the center of the valve, and a marginal ridge around the edge of the valve itself, and a flange on the mantle rather than a siphio marginalis. Okay, so there are, we talked a little bit about the ocellus being monophyletic. And when I say the ocellus is monophyletic, I mean the ocellus as it's defined by the Eupodiscales. That again, convex sheet where that is elevated and has the distinctive rim around it, which contains all the pores in the pore field. So you'll see a number of taxa in the mediophytes and beyond that have similar structures, but they aren't really ocelli. So things like Lampriscus, which does have a rim around part of the pore field, the apical pore field. So it's really more like an interrupted pseudocellus. You'll see a similar structure in some Glyphodesmus species. You see the ocellolimbus in some Araphid taxa, where this is actually the porous plate is sunken below the rim and the rest of the valve, as opposed to a convex plate grading directly into the hyaline rim. And then, of course, in the Somatocyrales, we've got a structure called an ocellus. This can be slightly elevated. Rather than being convex, they are concave. So again, they're sunken. So again, an ocellus-like structure, but not what we would consider a Eupodiscalean ocellus. Okay, oh, it's probably well past time that I took a breath here. I was almost reluctant to include a list of people to thank, because I've had so much help collecting and identifying these Biddulphioid taxa over the years. But here are a few of the people that I owe a great debt to in putting some of this work together. Well, it looks like I might have some time to take questions at the end here, but if you have anything else, you are free to email me. There's my address, and yeah, happy to take any questions.
Thank you very much, Matt. I really appreciate your presentation today, and yeah, truly showing us one of the most beautiful groups of diatoms. They just are the most photogenic, and you think you see in everything, and you turn the corner on them, and there's something else you've never seen before on them. So thank you.
Yeah, and I didn't have to cheat by going into the fossil forms. There's a tremendous fossil diversity with these groups that some structures just beyond description. I'm not even 100% sure how these work. Linking spines that wrap around each other.
Oh my.
And I don't even know how that evolves, how that grows. But
all right. Well, thanks again, Matt. If people would like to ask questions, please feel free to either put your question in the chat, or if you'd like to just go to the raise your hand feature at the bottom of your screen, and we can have you unmute and have you ask the question in person. As we wait for a few questions to come in, Kyle said pictures did not disappoint. Thanks, Matt. A great tour of the Biddulphioid diatoms. I have just a couple questions. You thanked a whole bunch of people that have helped you out, and I just was very curious if most of this work that you've been doing has been based on collections, museum collections, or if you're actually going in to culture these, and if you are culturing them, how are they in culture?
So it started out mostly with culturing. So a lot of them didn't exist in culture collections when I started my PhD work. So most of it was going out and getting them. The dirty little secret about collecting these guys is that they tend to grow in gorgeous places. So tropic, subtropical, sandy beaches. It's real real tough work. We can't all be drilling cores through the ice to collect our diatoms. But as this has expanded, obviously I've started to do a lot more work with historical collections, trying to integrate some of the fossil taxa, because there's been a lot of extinction in these groups. And if you want a big picture of the systematics and classifications of the Biddulphioids and really the whole mediophyte diatoms, you really have to look at the fossils as well. And thankfully, there's a lot of morphology to look at, as you can see. So,
and in the fossil record, do these? I picture these as sort of more shallow water nearshore forms. Do you get good representation of these groups in the fossil record?
There's a smattering. I don't want to. I'm still learning about the stratigraphy, so I don't want to say too much here, because it might be just based on my own ignorance as opposed to anything else. But yeah, there are quite a few out there, and some of them can be locally abundant. Some of them are great in terms of the culturability. Some of them are great lab rats.
Nice.
Diatom in particular, that grows like a weed. So
neat. Layla's got a question. If you'd like to unmute yourself, Layla, go ahead and you can ask your question.
Hello, thank you, Matt, for this. It was really very interesting. And I wanted to ask you if you use only light microscope and in fossil record, and you have just a fragment. How do you distinguish Biddulphia from Terpsinoe? Thank you.
So typically you can see the costae on the surface of a Terpsinoe, even in the light microscope. So that's actually one of the reasons that I'm in London right now, is to look at some of these fossil taxa, and you do see a lot of fossil taxa identified as mostly anomalous. I'm seeing that when you get a good look, you can see those external costae, and you realize that it is actually a Terpsinoe. The Terpsinoe also tend to have very flat valve faces, so you don't get the real elevations that you get in Biddulphia sensu stricto.
In fact, sometimes I don't see the costa, because when we see them, we recognize, of course, Terpsinoe, but when it's just a fragment of areola, it's a little bit difficult with light microscopy.
It can be, and I don't mean the internal costae. I'm talking about the external costae. And with some really fine microscope work, if you try to focus on the surface of the valve itself, sometimes you can see that network-like structure of the external costae. I don't want to say always, because obviously there's fossil taxa that are, fossil specimens that are degraded, but that would be the first thing that I would look for.
But in terms of ecology, is it very, very different if we are working on littoral core?
I think, and again, I don't know that much about that. I'm still learning about the fossil forms. I think Terpsinoe is primarily freshwater, but no. And I'm not counting Terpsinoe americana here, which I think is another beast entirely.
Oh, I have Terpsinoe musica.
Musica in a marine environment?
Yeah, in Mediterranean, yeah.
Oh, that's interesting. I'd be interested in learning more about where that came from.
In fossil record, I have it for the moment.
Cool.
Yeah. In quaternary, just quaternary record, but I can send you a photo of it if you wish.
Thank you.
Okay, thank you.
Yeah. Thanks, Layla. Matt, when you showed the picture of that Terpsinoe americana, I actually haven't seen it. Is it extant or is it a fossil form?
It's extant.
Is it?
So what I showed you was pictures from a culture from the Savannah River Delta.
Mm-hmm.
I know Andre Witkowski has seen it I don't know where he's collected it, but it does exist. It is extant. It is out there.
It looks very much like a fossil form when you see a picture of it. Matt, you introduced a taxon that I think you and Ed described, a genus that you and Ed described. It was Triaries. And where did that used to be placed before you guys erected a new genus for it?
So that's been in Biddulphia and Odontella. So Biddulphia mobiliensis, Odontella regia, Odontella or Biddulphia sinensis or chinensis.
Okay.
So it was that group there.
All right, thanks. Yeah, anyone feel free to add some questions to the chat if you have any, or go ahead and raise your hand. One of the diatoms you showed, Matt, was a Biddulphia sculpta. You talked very clearly about the annulus being round in some and elongate in others. That Biddulphia sculpta seemed to have some very strange structures in the middle that were almost like it had 3 annuli in there. Is that just an artifact that I'm seeing, or is that actually potentially something that is really different?
It's there, and it actually identifies the species. So I think it was either Brightwell or Shadbolt that described the species, and he called them orbiculi. I think the three orbiculi. Whether or not they are annuli or something else, I don't know. I've been trying to catch morphogenesis in that species. But yeah, that's a great question. And if someone has spotted them in morphogenesis, please let me know, because yeah, it's a structure that I'm interested in as well.
Well, mostly I'm excited that a diatomist actually gave the structure a name, because we never do that.
Well, it was named and then sort of promptly forgotten. Right?
But at least they named it,
Sure.
because yeah, we often get to talk about that, how when we see something on a diatom, we gotta come up with a name for it, and
Right.
we do a good job of it. So no complaints. Well, let me see if we have any other questions coming in here. I don't see any in the chat. So I think we'll start winding down here, Matt. Again, I really appreciate your presentation today. I do want to bring up one last thing. You mentioned several times the Iowa class. Each year since 1963, we've been teaching a class at the University of Iowa and Iowa State Field Station, which is called Iowa Lakeside Laboratory. And we are again hoping to teach the class this year. Myself and Dr. Sylvia Lee will be the professors in the class. The class is going to run May 23rd to June 17th this year. And we are hoping maybe to see if we can actually have a virtual component of that class as well. Please feel free to reach out to myself or Sylvia and for details, and we can also bring you up to speed whether we're going to be able to pull off a virtual aspect of it this year. We're actually meeting later this week to see if it's going to be possible. With that, let me do a quick check, see if we have any final questions. Look, I don't see any in the chat right now. I sure appreciate everyone joining us today for another episode of the Diatom Web Academy. Again, within 3 days to a week, we'll have this recording of this posted up on our YouTube channel, which you can access at diatoms.org/news. And mostly, I want to thank you, Matt, for giving us a tour of some of the most charismatic diatoms that exist out there. I surely can understand why you are intrigued and interested in them, because they look like there is no end to the surprises that they hold.
Yeah, well, thank you all for giving me the opportunity to ramble.
All right. Thanks, everyone. We'll see you in 2 weeks. Again, March 15th, Joe Mohan's going to be talking about three-dimensional applications in diatom ecology and paleoecology. All right, thanks again, everyone, and have a good rest of your week.